to go to a particular gene, the one that you want to. So, and then these are, you don't have to know all the details, but this gives you a pretty good idea of kind of the description of them, kind of the compare and contrast aspects of them, okay? Now, of course, the zinc finger nucleases and talons aren't naturally occurring proteins. These were recombinant proteins. They were engineered just taking two different parts of entirely different genes, putting them together. One part would provide the DNA binding specificity, the other part would provide the nucleus. CRISPR-Cas is, of course, as you all know, I think, from a naturally occurring system discovered in bacteria. And I do want you to know a little bit about that naturally occurring system. This just gives you some more ideas about the different targeting systems that can be used. This shows you more diagrams of the zinc finger nucleases and the talons. In both cases, the actual nuclease activity, the digestion of DNA, is by a restriction enzyme. But this is what I think this is the only restriction enzyme that I know of that doesn't have any target specificity, which is really bizarre. But what they realized was that, hey, let's take advantage of that non-specific nuclease activity by POC1 and make a recombinant protein where we're adding either zinc fingers or the talons to provide the DNA binding specificity. Okay, very clever. And there are still people that are using this because there are, in many ways, they can be more specific even than CRISPR. The problem is that they can be very difficult to engineer. You basically have to use some, computational tools to make predictions about what would be the amino acid sequences in your zinc finger domains and in these other domains here with the talons to engineer them to be specific, and then you have to test some of them, and they can be kind of expensive. Okay, so yes.
So, what would be like the difference, like the main difference between the two, or are they just both like generically the same?
They're pretty much the same, other than the fact what's guiding the DNA binding specificity. Now, the CRISPR-Cas system, if you don't know about it already, I just wanted to spend a few minutes talking about it because I think it's useful to know what it's actually derived from. And any of you that are in the Kearns lab, as I said the other day, correct me if I say anything wrong. But now the actual locus, the CRISPR locus, had been identified in bacteria, a while back, but people didn't have any idea what it was doing. They just saw it as a region of the genome that had these short repeats that they couldn't figure out what was going on. But then it was discovered that this is actually part of what's called an adaptive immunity system in bacteria to protect themselves against invading DNA, and that invading DNA is typically coming from phages, bacteria phages, right? So those phages attach to the surface of bacterial cells, and they literally kind of inject their DNA genome into the cells, and then they end up that the viral genome expresses viral genes that take over the machinery of the cell, and while new viral genomes are being replicated, they get put into a capsule once the viral capsid proteins are produced, and there's so many of them, and there's actually an active lysis mechanism that's encoded by the phage that just bursts open the cells, causing cell death. So the virus doesn't care, it just wants to use the host machinery, and then it moves on to infect the next bazillion cells. Okay. Well, what the bacteria of all the system, so that in their genome, they store little remnants of viral DNA, but specific for different bacteriophages, okay? So then when they get a live cell gets invaded, that DNA from the phage enters into the host cell, and because of this CRISPR adaptive immunity, what that DNA from the virus does is just get chopped up into small pieces instead of being able to express the viral genes and take over the cell. So it's a really cool. Now, this adaptive immunity system doesn't have anything to do with the immune system of vertebrates, right? There's no antibodies there. It's entirely different, but it's still a form of immunity. So I took this from a review article from one of the main labs that had discovered this. So there's actually 3 different phases, the adaptation. But what happens here is, let's say there's a virus, a phage that infects, okay? And during the course of that first infection, what happens is that this DNA is recognized and integrated into what is now known to be this CRISPR array. So just short chunks of the phage DNA. It doesn't matter if there's a gene, a viral gene there or not. It's just short chunks of the DNA. Well, this array could have, lots of different DNAs from different phage genomes, but it's specifically located right downstream from this operon that expresses several different genes, the Cas genes. Okay, so the array is actually CRISPR, okay, and Cas is the name for the genes that are expressed from there. Well, and look, that little arrow, the CRISPR array gets transcribed. So that's a gene now, even though this is kind of assembled by the bacterial cell, it gets transcribed. And all of these little guys, they have these specific intramolecular hydrogen bonding, forming these stem loops. And that's the basis, the stem loop formation is how Cas would be expressed from, I think it's this open reading frame. Cas protein recognizes these stem loops and it knows to process it. And there's some other proteins and nucleases involved in this processing, but you don't have to know about it. Okay, so we've got this mature cRNA that's interacting with Cas. What that Cas and RNA complex does is then just search for homology between this array, the processed RNA, and any newly invading viral DNA. And it makes a double-strand break. So you have to have the stem loop formation in the CRISPR RNA. You have to have Cas9. And you have to have base pairing between complementary base pairing and note that it's just one strand of the target DNA. Okay? But once that recognition is made, that complementary base pairing between the CRISPR RNA and the target, you get double strand cleavage. Yes?
Does the CR in cRNA stand for cellular ribonuclease?
Probably, or CRISPR, I'm not sure. Yeah.
Okay.
Is it CRISPR just CR? Okay. Are you in the Terns lab also? There's 3 of you?
Yeah.
Okay.
Okay.
So now this is kind of a generic system, and as the people in the Terns lab, or my Terns could tell you a lot more, there's actually different versions of the system, type 1, type 2, type 3. Don't worry too much about them, different kinds of prokaryotes. But what CRISPR-Cas genome editing is based on is what's called the type 2. Okay, so they're based on different nucleases. In this particular case, the Cas9 nuclease. Now, the other thing that's not on the previous slide that's important when you're thinking about the adaptive immunity system in bacteria is this other RNA called tracer RNA. That's a gene, tracer RNA. So with the type 2 CRISPR, you need to have not only those cRNAs that are made from the array and Cas9 being expressed, you also have to have transcription of this tracer RNA. So there's actually 2 RNAs involved that are required for this type 2 adaptive immunity system. And the tracer RNA is actually really, really critical because without the tracer RNA, Cas9 is not able to find and process or even target the CRISPR RNA to the viral DNA. In the cell, in the naturally occurring adaptive immunity system, you have to have tracer RNA. Okay? And don't worry about all these processing steps, that's for aficionados, but there are a number of processing. Now the other thing that's shown here that's really important, my batteries are getting down, it's really important is that PAM sequence. Not named for a particular person named Pam, but named for Pam, I have it on one of the slides, I can never remember, proto-adjacent motif, proto-spacer adjacent motif. Anyway, the importance of it is this 3 nucleotides. It has to be in the genome, in the DNA that you're targeting, and that's one of the limitations of CRISPR-Cas9. If the sequence you're trying to target in the genome doesn't have a PAM sequence, you're not going to be able to target. Now the nice thing, because of scientists doing so much extensive research has been done on CRISPR and the other Cas9 proteins in other organisms, multiple organisms. Now all kinds of, probably hundreds of different prokaryotes have now been identified to have different versions of this. Well, they've identified different Cas9 proteins or different Cas proteins that have different target specificity that depend on different PAM sequences. So that's not nearly the limitation that it was initially, okay, in terms of your targeting. And they're developing a whole lot more flexibility into this system all the time, because as people realize quickly early on, that was a limitation. But notice what I say here. One of the most important research papers, who are Watson and Crick? Why is Watson and Crick paper considered to be so important? Double helix model. I would argue, I've never heard anybody else say this, I would argue that this 2012 paper from Jennifer Doudna's lab is probably the most significant since the Watson and Crick paper, because now I took this figure from there, what her lab realized is that one of the limitations of using tracer RNA is that that's just a second RNA that you have to provide to the system. Okay, so 2 separate RNAs. One, you would have an RNA that you would either have transcribed from a plasmid, from an expression vector, or you would have it synthesize the sequence that you want, because you've designed that to be complementary to your target sequence. You would also have that make sure that region that you're targeting has a PAM sequence, okay? But the problem with using the naturally occurring system is it'd be a pain in the butt to have to add the second RNA in there. Well, what the Doudna's lab did with this 2012 paper, I have the reference on the previous slide, they showed that they can make an artificial chimeric RNA that basically performs the function of the tracer RNA. And they call that the single guide RNA. Single guide RNA instead of having 2 RNAs, okay? So that's the beauty of the system. It's much easier to just have the Cas9 protein, and that can be expressed if you're doing genome editing of cells like in culture. You have to have an expression vector that will express Cas9, or you can inject Cas9 protein in or inject Cas9 RNA, but typically I think there's an expression vector. You have to have your single guide RNA. Now notice the important components of it. One part of this guide RNA has the homology to your target, the gene you're trying to edit, okay? And there is a PAM sequence. This part of it would be common to every editing RNA that you would be using, and that's forming this specific secondary structure that gets recognized by Cas9. Okay. So that was the beauty of it, and this is just another figure that I took from another review article. It's not signed, but just telling you a little bit more. Now that PAM sequence is 3 nucleotides. And you think, well, just on average, any given 3 nucleotides, especially when one of them can be any of the 4, okay, that's great. But that still doesn't have a limitation. If you wanted to make, instead of making edit here, that's, just 10 nucleotides away from the PAM sequence, if you wanted to make the edit over here, 20 nucleotides away, you have limitations. You can only have a certain length of the targeting RNA, and it can only be a certain distance from the PAM sequence. So that was a limitation. But now we know there's different species of bacteria that have different PAMs, okay? But the point here is that you make this synthetic guide RNA. And the nice thing too is that you could have 10 different guide RNAs. They would all have the equivalent of the tracer RNA, the functional equivalent of the tracer RNA, but each of the 20 would be different in sequence for your gene. You could target 20 different genes at the same time. You could target 20 different regions of the same gene, and then pick out the one that worked the best. Don't worry, you don't have to know any of these. I just want to give you an example, and this is from that review article to decide, just all the different kinds of PAM sequences and the different nucleases that have been identified that can be used. So this has really greatly expanded the use, more flexible use of the whole CRISPR-Cas9. So I took this from an article that's not assigned, but you just need to know a couple things about this. You just have to introduce into your cells whatever you're trying to engineer. Now remember, these could be cells in culture. But even beyond that, what's so cool is that you can do it in oocytes of any organism. People are using CRISPR on mosquitoes to make gene drive. They're doing it with fertilized embryos of humans. Now IVF culture, of course. Other than the Chinese scientists doing germline CRISPR and having those babies being born, there shouldn't be any other, and that's obviously one of the debate topics. But has anybody heard of a particular CRISPR success that happened on this campus just recently, a couple weeks ago? Yeah.
I don't know what they did, but it was a lizard.
It was a lizard. Yeah, it's the first time CRISPR has been used for squammy, a lizard. And what they did was they did CRISPR gene editing of the tyrosinase gene. And tyrosinase is the gene that when it has mutations is responsible for albinism, lack of pigment in every organism. And I don't think they were expecting to get bi-allelic editing, but it apparently was a lot more efficient because they almost immediately got some albino lizards. And it's a pretty big deal. But, so that's the kind of thing that they did that by injecting it either, I think it was into the oocytes. I'm pretty sure, was it oocytes? Yeah. Are you in that lab? Or
No, I haven't.
Yeah. So, but anyway, so you have to have the guide RNA being expressed, or synthetic RNAs, and you have to introduce Cas9. Of course, if these are eukaryotic genome that you're trying to engineer, it's not going to have any Cas9, of course, but you can easily put that on an expression vector or plasmid, or you can introduce the mRNA that would get translated. You could even introduce the protein. So it just depends on your system, your cells, what works the best. So those are the three possibilities. Now, this is from the assigned review article. So here's the three different targeting strategies, but they all result in a double-strand break. And then there's the choices: non-homologous end joining, homology-directed repair. Okay. So this can be used just strictly in the absence of adding exogenous DNA for the repair. Okay, look back on the slide a couple slides back. This is going to result in deletions. Why are we getting deletions here? And why are they not the same? I thought we were just making a double-strand break there. This looks like there's more sequences. Double-strand break should be just across on each strand. Why are we getting deletions here? Non-homologous end joining. Anybody remember anything about that repair mechanism? the way it jams those two broken ends together is it's got to shoo off some of the sequences at the end. Now, this is the thing that can't, at least as far as I know, can't really be controlled very well, but let's say we're gonna be talking about doing this in pluripotent stem cells in culture. So you'd introduce your Cas9 with your guide RNA into the cells. You're going to get, like we talked about with EZH2 paper in January, okay? But you'll get clones of cells that you just have to go through and screen for any of them that have deletions, and then you just sequence them and identify the ones that you want. So in some cases, you might want to just have a deletion of a couple nucleotides. Or you might get deletions where they're in frame, and you don't want that if you want to make a knockout, right? Because you want, I mean, yeah, it's possible that an in-frame deletion could knock out the protein, but generally, you want it to be out of frame just to make it more likely that it would be a non-functional protein. Yes.
So does the non-homologous android have like potential unintended downstream effects for like other genes or?
Okay, that's a good question. It's not the NHEJ itself. This is the naturally occurring DNA repair system, and it's used in eukaryotes most of the time to repair double-strand breaks by like X-ray irradiation. The downstream, unintended downstream effects are more having to do with the lack of specificity of any of these three targeted events. So they make double-strand breaks someplace else.
Right, because they might, if they're looking for a certain sequence, they might find it.
They might find something that, maybe there's something that differs by just a nucleotide, but it's in a different gene. And the problem is, now they've got some predictions about how to search for those off-target mutations, but it's still one of the big concerns about CRISPR genome editing. Yes?
Could it be an endo and then like downstream enhancer?
Yeah, you can do this anywhere. It could be in a coding region, it could be in an answer sequence. Yeah, I was referring to in frame and out of frame with coding sequences, but you can do exactly the same thing with an enhancer, with something that you think is like an insulator or any kind of cis-active regulatory sequence.
I mean, could like protein coding sequence be an enhancer?
And a protein coding sequence. Well, yeah, you can have sequences in one gene that even if it's coding sequence in that gene, and there are sequences that can act as an enhancer for another gene. Usually they're in introns of other genes, but it could be. So this is a way of making deletions, and then you just screen. This is just showing, one strand or one homolog each, but you look for the ones that you want. Now, if you want to make, say, point mutations, either mutating a wild type allele from cells that have the wild type, say you've isolated iPSC cells from a patient that doesn't have a genetic disorder, you can convert that to wild type, the disease-causing wild type, and you can do the same thing. You can get cells from patients that have the genetic disorder, make iPSC cells from them, and then correct the mutation. And you get matched isogenic. The term is isogenic, meaning if you've got a wild type that you converted to mutation, those two cell lines, the original and the mutated one, are everywhere else in the genome are exactly identical, isogenic, with a possible problem of off-target effects. The same thing, and this is the beauty of disease modeling with iPSC cells. You take the iPSC cells from the patient that has a genetic disorder, correct that mutation again. You've got two isogenic cell lines that are matched everywhere else in the genome except for that one mutation. Now, the cool thing too is that then we're not going to get into, I'll just mention it in brief. So they've been able to, there's just some amazing stuff going on in terms of studying how, like structure function of Cas9 to determine what exactly it is about the sequences of Cas9 that make it function like this. But the nice thing about that basic research is they've also been able to engineer the original Cas9 for doing all kinds of other things other than just making double-strand breaks. There are mutations that have been engineered in the Cas9 that make them do nicks instead of, so just nicking one strand instead of making double-strand breaks. There are other experiments where they make chimeric proteins that still take advantage of Cas9's ability to interact with guide RNA and guide it to the target. But instead of it cutting that DNA, the Cas9, the nuclease activity of the Cas has been knocked out. So no nuclease activity, no cutting of the DNA, but still retaining the ability of Cas9 to target a specific sequence. But then they add a domain for like histone modification, the catalytic domain of a histone modifier, so you can target, say, a histone deacetylase to a specific enhancer or promoter and use the targeting capability of Cas9 and then the histone deacetylase activity and that kind of to target there. Okay. And the methylases for DNA, there's just all kinds, they're even adding small molecules like fluorescent molecules, just all kinds of people are just amazingly clever sometimes, I think. We will talk about two other applications beyond just making knockouts. One of them will be actually, so the first paper we'll talk about with the muscular dystrophy paper, they're not making small deletions, they're making ginormous deletions. All as a way of partially restoring the function of the dystrophin gene product. It's a huge deletion. Okay, we'll get to that. They had to use 2 guide RNAs, and it was a little tricky at the time, and it's kind of 2016, and the technology between 2016 and now has, because of people like the Jerusalem studying more things about Cas9 or Cas protein, the technology has just exploded in the field. Another thing we'll talk about with respect to sickle cell and other blood disorders, is you can actually even do screens using CRISPR. So you can screen for mutations that affect the cell function. So using CRISPR as a mutagenesis tool, random, genome-wide. And this was really a spectacularly successful one that we're going to talk about. Okay. And then we'll talk about the last set of papers we'll be actually talking about slightly different applications. So this is just giving you a little bit more from that assigned review article. Again, just starting with a double-strand break and the 2 possibilities. For this possibility, you have to add, you can add an oligonucleotide. I'd say for a point mutation or a point substitute, single nucleotide substitution, you could add a larger fragment of DNA. It's just got to have homology to the region that you're targeting, and then whether it's in the oligonucleotide or the double-stranded fragment of DNA, you introduced into this DNA, the donor DNA, the whatever sequence alteration you want to do. And this homology-directed repair is actually surprisingly efficient. You still have to screen for it, but with next-gen sequencing, it's really fast. And these are just some of the things that are being used. People are even using this to, let's say you asked the question about enhancers. Let's say you've got, a region of a genome that you're pretty sure has an enhancer function, but it's a pretty big region. Maybe you've only got to narrow it down to, a 5 kb region or something. But what you want to do is go through and systematically identify, dissect that region and identify the exact sequences. Well, you can do that with site-directed mutagenesis, but it's still very tedious, and you have to do it with transgenic organisms. But they can actually design. Remember how I said you can use one? You can introduce 20 different guide RNAs into your cells, along with the Cas9. But what you can do is have each of the different guide RNAs, along a 5 kb region. You might target here, 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 just systematically mutating every individual region in whatever region you're trying to test that. And then just screen the ones that are affected, and that's done in cells. We'll even talk briefly next week about not only is the technology being used for germline modifications of a lot of experimental and natural populations, it's being used, you can actually introduce Cas9 and CRISPR, the guide RNA, in vivo. Now, it's still not to the point of being in humans, but basically doing in vivo gene therapy but involving CRISPR. And this is a figure from, it's not exactly like the signed one, but it's from the same author, just from a few, I just like this figure a little bit better, but this is what I was talking about a few minutes ago about, you can take, there's a lot of studies still being done where you take cases and controls, okay, and different individuals, you might have, 5 people that don't have the disorder and 5 people that do have the disorder, and you make iPSCs from each of them, and then compare some properties or molecular characteristics of the mutant versus wild type cells. Well, you can't, if they're not isogenic, you can't determine exactly what would be different between the cells because they're not isogenic. Okay, it's still useful, right? Because then you are representing the genetic heterogeneity that's present in humans, right? You want to know on different genetic backgrounds, does a particular mutation that's causing a disease have the same molecular or cellular effect, okay? But it's difficult to tease out if they're non-isogenic. On the other hand, you take wild type, convert it to mutant, or you take mutant and convert it to wild type. That's a more control, that's like doing, gene targeting in inbred strains of mice, okay, because you've got them being isogenic. So there's a lot of really nice things that are being done. So these are some of the issues, off-target effects. You never know, as I mentioned the other day with iPSCs or anything that you put in culture, you never know for sure if some other thing hasn't happened at the chromosome level. There's always a possibility if you're doing any kind of cell-based therapy, they can become oncogenic. Can you get enough? I think this is what Shinwa Yamanaka was referring to in the video the other day. Now, yes, this is really useful for monogenic disorders, but can we get any information from polygenic? And yes, we're getting that, but it's just more time-consuming. But it will happen. So, we've got 2 different kind of approaches. Regenerative medicine. You take those iPSC cells from a patient that has a genetic disorder, you correct the mutation, those cells you can transplant back into the patient, and there's no post-immune reaction against them because they're allogenic, or autologous. Allogeneic is from different individuals, but as Yamanaka talked about the other day, they're in the process of engineering, changing, or identifying individuals, iPSCs from individuals that have different HLA haplotypes, and finding what he calls to be super donors. So basically just controlling for the HLA haplotype, genotype, and then they can use these iPSC cells for lots of different people. Okay, so if you weren't here Tuesday, I would strongly encourage you to watch that video. Not so much for things that you'll be tested on, but things that I think you'd find very interesting. So that's regenerative medicine or cell-based therapy. We can also use, and this is what I'm going to be focusing on with the papers I've assigned, is understanding the disease mechanisms. Because you can take one set of iPSC cells from one individual, or you can have control and mutant. Differentiate, remember iPSC cells, you can control the differentiation into lots of different cell types, you can take the same cells, iPSC cells, program them into skeletal muscle, program them into cardiac muscle, program them into blood cells, program them into neurons, whatever you want, and then study the, as I said, the molecular and cellular. They're even being used for drug screening, which is really very powerful too. And this is just what I want you to think about. I mean, all these different things, drug discovery, developmental biology. People are taking iPSC cells and doing these three-dimensional cultures and growing organoids that really are differentiating. They're forming specialized layers of both of the brain and the heart, even embryos. They've been able to culture embryos derived from iPSC cells, and they put them under these specific 3D cultures, and they start forming all the 3D germ layers. It's really, really cool stuff. You can learn a lot from that. Drug discovery, cell-replicative disease mechanism. So, just based on what your, kind of what your own general interests are, what would you want to do? Would you be more inclined to go towards drug discovery, using, making iPSC cells from people that have different alleles of a particular disease gene and comparing them against isogenic controls, and then just testing, 20 different drugs for what effect. Do they kill all of the cells? Is there one out of those 20 that doesn't kill them and it makes them do something that you want them to be doing? Yes, you can screen them. It's much cheaper than animal models, much more valid than animal models, avoids the controversy some people have about using animals for drug testing, right? Anybody have any initial thoughts on what would be most appealing to you? The more basic stuff, disease mechanism or developmental biology, or direct clinical application, translational medicine. Translational medicine. Anybody else? Nobody's going to say basic disease mechanisms or basic developmental mechanisms. Come on. That'll be another quiz question. What do you
I
say?
think developmental biology is very interesting.
I agree, but I would like all of them too. I used to work for a pharmaceutical company, not specifically drug discovery, but I do like developmental biology and disease mechanisms better. This is what I was saying, you can do this with human embryos. And there are 2 papers. And there's been I haven't looked to see, there's been some controversy whether the claims of these, now these were not the ones that were germlined by the Chinese entity. I think it was an entity. These are ones where these embryos were just put in culture. Now the problem with this, and I don't know if it's ever going to get resolved, at least in this country, there's a number of years ago, I don't even know who originally came up with it, but most countries follow what's called the 14-day rule. With human embryos, you can't culture them past the equivalent stage of development that would be 14 days, 2 weeks in utero. You can't do it. So there's a lot of things that happen after 2 weeks, though, that a lot of people would like to be able to study further with embryo cultures like this, especially to being able to do genome editing. Yes?
Is the 14-day rule like a legal rule, or is that just like an agreed upon?
It's an agreed upon ethical rule. I don't really know. I think that's a good question. I don't know the details, but I would suspect if somebody tried to do it here, they would lose their funding. I don't think that they'd be thrown in jail or anything. Yeah. But for a university scientist, and all of this would be NIH funding, and to lose all of your funding, yeah, you're not going to break that rule. Now, if a pharmaceutical company or some other company tried doing it, there could be legal sanctions against them. I'm not sure. Somebody look that up. That's a good question. Yeah. And as far as I know, that 14-day rule is followed by a lot of countries, and there's not anything to say that it'd be all countries. Okay, and just so you know, I think this site is still active. You can go there if you're interested in any. This is a little bit out of date. but you could go to this website and find more current. And this is a really good Jennifer Doudna. It's ancient now, 4 years ago, but it's still a really good review. And of course, there's this problem, the slippery slope of germline editing. And we'll take a break with that, and then we'll talk about muscular